Hello there, it's Luke, and in this tutorial about haemostasis and coagulation, we're going to be turning our attention to looking at the cells within the wall of the blood vessel, looking at the endothelial cells and the subendothelial cells, and in doing so we're going to be considering what we refer to as the tissue factor pathway, or initiation if we're thinking about the cell-based model, or the extrinsic pathway if we're thinking about the sort of classical model of coagulation. We're going to be using our diagram again, so I'll draw your attention to the platelets, to the endothelial cells, which will be the sort of inner lining of the blood vessel, the subendothelial tissue, the sort of connective tissue, the sort of other cells within the wall of the blood vessel, and the clotting factors that are going to be circulating within the blood. So here is what it looks like in real life in cross section. So this is our blood vessel. Here's some endothelial cells. There's some red blood cells in the middle, within the lumen of the vessel. And this is what it looks like on our diagram. Okay, so this is the lumen, this white area here. We've got some red blood cells, some platelets, some white blood cells, and we've got the inner lining of the blood vessel. So the endothelial cells, and then all the subendothelial tissue and cells and connective tissue and what have you behind it in the wall of the vessel. And the first thing to draw your attention to is that the endothelial cells release nitric oxide and prostacyclin, and these are both vasoactive substances, and what they do is they vasodilate the blood vessel, so they keep it nice and relaxed and nice and open and reduce the resistance to flow so that blood can flow easily through that blood vessel. They also repel platelets, stop them sticking to the walls of the blood vessel. Now, it's important to note, especially when we're considering different drugs that we might use to prevent clotting, that prostacyclin is produced from the cell membrane, from phospholipids in the cell membrane, via something called the arachidonic acid pathway, and that will become more pertinent and more interesting to you when you begin to think about drugs that act upon the coagulation cascade. I shall say no more about it now. Now here we have a blood vessel with that's damaged. Okay, we've got a disruption of the blood vessel, and so we've got an area where these cells are either damaged or just not there anymore. They've been broken and damaged and aren't functioning, and so we don't have quite so much nitric oxide or prostacyclin being produced, and so consequently we get vasoconstriction, and the walls of the vessel sort of begin to come together. And when we consider our sort of bigger diagram, so we have an injury to the blood vessel, and we've got blood leaking out. Well, we're no longer producing quite so much nitric oxide, we're not producing as much prostacyclin, and so consequently that blood vessel's constricted, and in doing so it's not only has greater resistance to flow, so hopefully we're bleeding a little bit less, but also it's bringing a lot of the clotting factors that are dissolved in this blood, in the plasma, and the things like the platelets in closer contact so they can begin to interact with one another. The other really significant thing to talk about is something called tissue factor. Now, this is a clotting factor that resides within the membrane of the cells, within the walls of the blood vessel and sort of other tissues. So on our diagram at the top here, we've still got the lumen of the blood vessel through which we've got red cells and platelets and things flowing. Here's our endothelial layer of cells, and just behind that we've got these cells with tissue factor expressed on the membrane. And there's lots of different cells that have tissue factor in them. We're talking about these sort of subendothelial cells just now, but also leukocytes. Other cells within the wall of the blood vessel will express tissue factor, and I'll explain why that's significant. So we've got this injured blood vessel, and we're going to zoom right in on this area here, and you can see that because the blood vessel is damaged, the tissue factor that's in the membrane of some of these cells here is now exposed, and the blood's all sort of tumbling out, and within the blood we've got red blood cells, platelets, but we've also got clotting factors, and we've got some calcium in there as well. And tissue factor and factor 7 have a particular sort of strong relationship, if you like. They work together, and in the presence of calcium, they form a complex. And what that complex does is catalyse other enzymes. So it activates other clotting factors. And the clotting factors that it works on are factor 10, also known as Stewart-Power factor, and factor 9, which is known as Christmas factor. And this mechanism here of the tissue factor being exposed and interacting with factor 7 and then going on to catalyse 9 or 10 is known as the tissue factor pathway, or initiation if we're discussing, if we're thinking about a cell-based model of coagulation. And if we're thinking of the classical model of coagulation, we might refer to this as the extrinsic pathway. Now, what's significant about this reaction, whether it's 10 to 10A or 9 to 9A, is that then goes on to activate another enzyme, another important clotting factor, which is 2. And this is also known as prothrombin, and the active form is known as thrombin. And this is probably the most significant, if you like, factor within the whole coagulation cascade, in that it has lots of actions, and one of the principal actions it has is to activate platelets. And we'll go on in the next tutorial to discuss exactly what an activated platelet does, and then furthermore we'll discuss the many roles of thrombin as well. So in summary, we've talked about the role of endothelial cells. We've said that they produce vasodilating agents to keep the flow through the blood vessels. When they become injured, they produce less of those, and so the blood vessels constrict, which is helpful in stemming the flow of blood. When you have a damaged blood vessel, you expose tissue factor, and this sets off very rapidly a cascade of coagulation to help with haemostasis, that ultimately leads to the activation of prothrombin to thrombin, which is an incredibly helpful clotting factor, which will activate platelets and do many other things. Thank you very, very much for listening.